my name's Andrew. I'm one of the PhD students that's a part of Program 3, and I'm working on the bioactives for value adding to Artichokes project. And so my supervisors are Vicky Solar, Kirsty Bayliss from Murdoch University, and we're working with Julia and Mark McHenry from Mount Lindsay Farm. And they're the driving force behind this project, looking at how they can better utilize their artichoke crop to make more nutritious food for everyone in Australia. And so today I'm going to talk a little bit about the work we've done to try and optimize that, and particularly looking at ways of making them more suited to processing into value-add products. So I guess a little bit of background. So Mount Lindsay Farm down near Denmark is about 4 hours south of Perth, and it's not your typical farm. It's based in organic practices. And the artichoke crop that Julia and Mark have grown is grown from seed and offshoot propagation. And so the crop is incredibly, just move this so you can see. There we go. The crop is incredibly heterogeneous. So you get a whole range of different shapes and sizes and colors, as you can see in this picture. And while that might seem initially like a disadvantage, it's in fact a really positive thing about their crop in that they get a very long harvest period. So all the way through from August to November, they're getting a crop, whereas a lot of other farmers are seeing crops from July to August and then stopping. They also have a much larger range of potential genetics to work with to optimize their product. And so the reason that they want to work with artichokes is because artichokes are incredibly nutritious, as well as being quite a hardy plant that's perennial. So artichokes are excellent sources of phenolics, which you might know as antioxidants, particularly chlorogenic acid and dicaffeoquinic acid, which this project focused on. You might have heard those names before from coffee. It's also an excellent source of inulin, which is a prebiotic dietary fiber. Also has other dietary fibers such as pectins and lignans, and it's a good source of minerals such as potassium, iron, and zinc. And so naturally, we want more people to be eating artichokes as part of their nutritious diet. But one of the key problems with artichoke, specifically in Australia, is that they take a lot of preparation to prepare. So all those outer bracts, which look like petals, they need to be peeled off, then need to cut off the top half of the artichoke, and then you're left with about 30 to 40% of what you had at the start, which is actually edible. And so if you're a consumer and you pay $4.50 and you end up with 60% waste and you've spent 10 minutes preparing it, it's generally quite a disincentive for people to pick up that vegetable and cook with it. Not to mention that a lot of Australians don't know how to cook with artichoke. And so this makes artichoke a really good candidate for value-add processing, because if you take out all that labor preparation and that waste that the consumer has to deal with, you're going to create a more convenient product that they're more likely to incorporate into their diet. But some of the issues that we have with turning artichokes into value-add products, which have been explored before, is that they tend to go brown incredibly quickly. And so in this little video, you can see that we're already at 10 minutes and our artichokes have already gone brown after cutting them. And so enzymatic browning, which is caused by the enzyme polyphenol oxidase, essentially takes those phenolic compounds that make our artichokes so good for us, and turns them into brown pigments. And the general consensus has generally been that the more phenolics you've got, the more brown your product goes. So in that case, a lot of people have proposed that, well, if we want artichokes that don't go brown, so it's easier to process them, then we'll try and pick cultivars that have a very low phenolic content. But it's very counterintuitive that we don't want to take out that component of the artichokes because that's what makes them so good for us. And so this project was trying to look at, first, can we find artichoke lines on the farm that have got a really high phenolic content because they're more nutritious for us, but also ones that don't go as brown so we can get the best of both worlds. And so I'll just take you through some of these graphs at the bottom. So what we've done is we've collected 54 artichokes over 3 months of the harvest period last year. We've measured the color change as they go brown. So the L star, which is that bottom left graph, is showing you the lightness. So as they go darker, the value for L goes down. The A star in the middle is increasing, it's showing that there's an increase in the redness of the color. And the B star, which you can see is separate for each harvest point, which we'll get to in a little bit, is showing the yellowness. So there wasn't a huge amount of change in that sense. And then that final one at the end is showing you a total color change. And so one of the takeaways was across the different times that we harvested, we saw differences in the A and the B star, but overall, the amount of browning was the same on average. There were some differences between specific ones, but generally there's no big change. And so in addition to looking at the browning and the color change, we also looked at the phenolic content. And so we've analyzed the total phenolic content, so all of them together for the artichokes, and we've mapped them out to see what sort of variation we get. And what we found was a huge amount of variation across all our samples. So you can see the highest content that we got was 13.22 grams per kilo on a fresh weight basis, which was more than 3 times that of the lowest one that we found. And so naturally for Julia and Mark, trying to select artichokes that are the most nutritious to work with and to propagate, they're going to be looking down the left end of this graph. We did the same for chlorogenic acid, so one of the main phenolic compounds in artichoke. And again, we saw that more than 3-fold difference between the best and the worst artichoke lines. And the same for the dicaffeoquinic acid one. Now, you might notice some of the different colors. So we're looking at the early harvest, which is green, middle harvest, orange, late harvest is blue. And you can kind of see the general trend is the later we harvest, generally, the lower the phenolic content. And we found this was associated with the amount of solar radiation that the artichokes were getting. As we move towards summer, we get more solar radiation. I think what's quite interesting, quite valuable, is that you can see at the top end, we've got a couple of the mid and the late harvests, which have got relatively high contents of phenolics. And so they make really good candidates for propagation because you can still maintain your long harvest season, which is a competitive advantage, but you're still maximizing the nutritional value of your artichokes. And so we also wanted to look at how do these two relate. So we've measured all the we've measured the color parameters, we've measured the phenolic content. Are there any associations between these? So we did some correlation analysis and we found we found 3 key highlights to pull out. So one of them was that our color parameters, or the browning, was mainly caused by the dicaffeoquinic acid. Chlorogenic acid and the total phenolic content were still correlated with the browning, but not to the same extent as the dicaffeoquinic acid. And this sort of suggests that if there's a particular phenolic that's causing that browning, if you can select to reduce it, you can still maintain a high total phenolic content while still having a processing suitable cultivar at the end of it. We also found, which is contrary to a lot of the previous research, that the enzyme, polyphenol oxidase, the amount of activity that it had wasn't associated with any of the browning. And we sort of think this is because there was so much of it that was already there, there wasn't really a difference. And it's just catalyzing the reaction and off it goes. One of the more unexpected findings that we had was that our phenolics were all correlated with the color of the artichoke. And so you can see the little picture down the bottom. That's a picture of the phenolics that we've extracted from the artichoke. And so the B star value represents how yellow it is. And you can see the color of the phenolic extract is also yellow. And so it's suggesting that the more phenolics you've got in your artichoke, the more yellow the flesh is going to be before it goes brown. And so that's presented a couple of options. We could potentially look at developing some rapid in-field measurement techniques to estimate the phenolic content, which could help speed up that cultivar selection process. But it also potentially offers a market advantage to Julia and Mark. If they're breeding artichokes that have got a higher phenolic content than their competitors, and you can visually see that difference, it makes it a lot easier to sell that unique selling proposition to a consumer. And so just moving on again and going back to that dicaffeoylquinic acid, which was causing the browning, that particularly stood out as a potential route for changing not just Australian artichokes, but potentially more widely in the Mediterranean region where they're grown. So we want to know, does the dicaffeoylquinic acid actually contribute more to browning? And so we did a couple of in vitro tests to investigate this. And what we essentially did was we took those two phenolics, chlorogenic acid and dicaffeoylquinic acid, we mixed them together in different ratios, and we then added the enzyme from the artichokes and let it go brown, and then measured the color. And so you can see in this graph, we've got this line down here, which is just chlorogenic acid by itself. It's relatively low absorbance, not a lot of color to it. We look at the dicaffeoylquinic acid one, and it's a little hard to see, but we're looking at the yellow line, and you can see that not only is it much higher up at the same concentration, but you also get more absorbance at the longer wavelengths. And when we reverse this to what color we're actually seeing, this relates to the orange and the red colors, which are indicative of more intense browning. That blue line, if you're interested, is the combination of the two, but it's around equivalent to the two lines combined together. Now, we also wanted to see, does this apply in the artichokes? So we took some of that phenolic extract that you saw in the previous slide. We dried it, we reconstituted it, and we then added in either the chlorogenic acid or the dicaffeoylquinic acid. And you can see dicaffeoylquinic acid, when you add it to the artichoke, gives you a much larger and more intense color response in browning than if you add chlorogenic acid. And so this really indicates that if we can reduce the amount of dicaffeoylquinic acid in the artichokes, we're potentially going to be able to get lower browning while still maintaining a very high total phenolic content. And so our key takeaways from these two parts of the project were that we've been able to find artichoke lines to continue in breeding programs that have really high phenolic contents. We've now found for inch. We've also identified that the phenolics are related to the yellowness of the receptacle, so associated with a visual quality parameter that can be easily recognized by consumers and could be a potential market advantage that can be used. It's not going to let me. Yeah, there we go. We could also potentially develop some methods to help speed up that process in the field to identify which artichokes are suitable to carry on with instead of going through the more labor-intensive and expensive lab analytics. And we've also identified a specific phenolic that's related to the browning that, if we can reduce while maintaining a high overall phenolic content, may allow us to develop cultivars or varieties that are better suited for processing, but still offer that high nutritional value that we're looking to maintain. Yes. And I guess the next, looking forward, beyond just artichokes, this applies to a wide range of horticultural products. Can we apply this same approach of identifying specific phenolics to create apple lines that don't go brown, but have a high phenolic content, or bananas or mangoes? So I think there's a lot of potential to explore this sort of approach in creating processing-friendly horticultural lines, but are still really good for us. Thank you. There you go.